So now our next speaker is going to be Kmiec from Christina Delaware, US. And Eric is going to tell us about CRISPR-directed gene editing for head and neck cancer, a patient-centric approach. Take it away, Eric.
Very good. Thank you, Ayel. So thank the organizers for having me here today. I'm going to tell you a slightly different story than what we've been hearing so far. Lots of great technologies and outstanding progress, certainly in Oskan's talk, amazing. But I'm going to talk a little bit about a slightly different approach that we've used almost out of necessity. We don't have the resources that many other folks do. So we sort of started to think about how best can we have an impact on patients, because ultimately we are here not only for the development of excellent technologies and processes, but actually to help in patient suffering. And so I think that's kind of the approach we took. So about 10 years ago, a group of us came together in Christiana Care, which is a 14,000-person health system in northern Delaware, most famous for treating the past president of the United States. President Biden lives about 10 miles from this hospital. And we divided our mission into two: discovery and translational research, which I'm going to talk about today, but something that Jimmy mentioned yesterday, and I think there were some great questions yesterday. We actually have a whole education division which has a product on the market called CRISPR in a box, and it's licensed and distributed by Carolina Biologics. And it teaches kids to get more immersed in the field, not just do the exercise itself. So it really resonated with me what Jimmy had said. So yesterday people were talking about folks that had actually studied gene editing over a course of many years, and that's me. I'm not actually a technology guy, but I've studied more of the understanding of how certain tools cause cells to respond, and I think it was discussed that there is a response in the cells that causes gene editing itself. And so Channing Durr, who's a pancreatic cancer guy, actually created a slide at a meeting in Boston this year, and I loved it, and it kind of showed his roles as a function of the number of albums that were present during his career. And here are mine. These are some contributions, and it kind of scares me. I was also scared yesterday when Chandra showed some remarkable pioneering papers, and I did them as journal clubs. So, so it does make me feel old, and here's the last piece of it here. So I want to talk a little bit about a patient-centric approach, and this is not new to us. In 2019, ethicists and I wrote a piece for Scientific American that most of the breakthrough technologies are either too hard on patients, so they never reach patients, those from the underserved communities. So we started to think about that, and I think from our perspective, it became much more important to see what patients will really do. And sometimes the technology and the science surrounds us, but we don't actually think about the impact on the patient. And we heard yesterday from Jimmy that he almost gave up. It's a remarkable story because of the preparation he had to go through to receive the CRISPR drug. And it's a hero that he went through it. It obviously worked, and he looks great, and he's a remarkable speaker. But we asked patients what really causes them to drop out of clinical trials, what causes them not to want to go forward, and these are some of the common reasons. The side effects are very severe, and in many cases, the preparatory work to get the advanced therapy are so severe they stop. And so you notice patient numbers go from number 16 to number 4. Well, those other 12 patients stopped or didn't respond. And a lot of times it's not anything to do with the technology or the approach, it's actually to do with the physical being of the patient. So we started to think about how can we help here. And so having watched CRISPR evolve through the years, like many other tools, I sort of thought that maybe the best use of CRISPR, certainly in genetic medicine, is to disable the gene. And the concept of that is more about using CRISPR for what it really does. And we know from tremendous amount of work from many people, some in this room, that it's very, very effective in gene disablement. And the goal here really is to keep patients on their clinical trials. I don't see CRISPR in our hands, what I'm going to tell you about, as a unique monotherapy, although it can be used in that way. And some of our colleagues now are publishing papers where, in fact, it is quite in oncology, pretty good monotherapy. But more about the complementarity to existing therapies where patients have exhausted their options, chemotherapy, radiation, and even immunotherapy, where the side effects are so dramatic they just stop. About 60 to 70% of cancer patients, certainly for head and neck cancer, stop their therapies because they just get too sick and they give up. And so that's where we thought we could make a difference. And so what gene do we knock out? Well, this is actually a gene that's been in the literature for a long time. It's NRF2, and it's a master regulator that controls a lot of different pro-carcinogenic activities. For us, the most important one was actually drug resistance, because we felt there was a patient who was being treated with chemotherapy or radiation, and basically, if we could relieve the resistance that happens, or at least reduce the amount of chemotherapy or radiation, the patient would functionally get better, suffer less, and complete the trial. Now, it isn't a cure, it's not something that goes on, but in comparison to some of the other treatments that are going on, we thought that was the right place to go. So, one of the most important things about choosing this gene is to pick the right target. A lot of great technologies, a lot of great information and things, but if you pick the wrong gene on the wrong site, it's not going to work. So we thought that maybe NRF2 would be a good target, but it is not a good universal target. We chose cancers, and particularly ones you see highlighted here, where NRF2 is highly upregulated. And that's going to be important for the sustainability of the treatment I'm going to talk about toward the end. And we still don't quite know why that is, but we're getting better results than we thought initially, which of course is always really scary. So we chose head and neck cancer primarily because in that particular cancer, in talking to surgical oncologists who are our partners, not medical oncologists so much, but surgical oncologists, their view was that NRF2 is upregulated in a lot of oral cavity cancers and head and neck cancers. And I don't know if you know much about this particular cancer, but it is the #1 cancer with suicide in the world. So people get so depressed because of the cosmetic changes and the fact that sometimes their scapula is used to replace their jawbone when the surgeon resects the jawbone. We thought, here is a place we might actually be able to help, and not only in helping patients along, but getting to that particular cancer. So here's the gene. Thousands of people here all over the place seen these kind of diagrams before, so I'm not going to belabor the point. I will say that we chose our sites the old-fashioned way. We actually did the work. I'm very aware of the commercially available guide RNA selection methods. In our hands, for this particular gene, they have not been particularly effective. That doesn't mean they're not for other genes, but in this particular gene, it's not. And so exon 4 and exon 2 are our two target sites. And the reason we chose those two is because in exon 2, and I'll talk about this data in a second, there's a unique mutation which makes the knockout in exon 2 of NRF2 tumor-specific for lung cancer. Now, the problem with that is that that's only about 2 to 3% of lung cancer patients, squamous cell lung cancer patients get that. So we went ahead and developed it, a CRISPR that knocks only the NRF2 gene out in lung cancer cells of squamous cell carcinoma. Exon 4 sites were chosen because, incredibly, they never mutate. So all the way through the development of the tumors, these two sites do not mutate, which allows us to make a single drug for various indications. At least that's the hope going forward. This is a standard experimental procedure we use. It's not that different than most of you use. I will say this. We see a lot of, and we plot it this way too, editing efficiency. That doesn't really mean much to us. Effective editing efficiency, creation of frame shift mutations, because the editing efficiency, CRISPR is very effective. I think we can all agree in cell culture and even in animal models. But the effective mutation is what we really want, particularly when you're trying to disable a gene that is a master regulator or a transcription factor. So when we targeted NRF2 at exon 2 at a cycle R34G, this demonstrates an important part of just cell behavior. If we have a single mutation in a heterozygous state, we can knock out about 33% of the target. The reason it's 33 is in this particular adenocarcinoma, there are three copies of NRF2. So the numbers work really well. If we get to the homozygous knockout clone, we see complete knockout effectively, and in normal bronchial cancer cells, epithelial cells, it's inactive. So this really convinced us that this is pretty tumor specific, at least under these conditions. And then we did a very basic, boring experiment. So I took the clones that we had actually created, isolated them out so it's a pure culture, implanted them in a mouse, and then tested the growth of the tumor. This may sound funny to you because why don't you just take the tumor, stick it in a mouse, and inject the tumor? But we wanted to be sure that our NRF2 target was legitimately genetically solvable. So we did that experiment, injected it, and then watched the growth of the tumors. And as you can see on the top lines, that's the growth of an adenocarcinoma, growing very, very rapidly in the back of a mouse. The red line, or the orange line, is actually one in which the NRF2 gene is knocked out in 1 of 3 copies. Grows much slower, and then when we add cisplatin behind it, we see almost complete cessation of growth. And all those tumors are actually necrotic. I'll say another word about measuring oncology, and many of you know this. Tumor growth size is hard to measure because as tumors are dying, they actually accumulate fluid. And so it's really hard to know if it's a squishy tumor or it's solid or what the levels are. So you sort of have to take that with a grain of salt, how they're growing. But if they are separated statistically from the control animals, you can be kind of confident you're in the right space. So we next did the experiment that everybody wants to see, implanted tumors in the back of a mouse, injected with the LNP, and we use a non-viral delivery system that we have built through about a year worth of screening of these lipids. Finally packaged it up, injected it, and we saw a pretty good distinction between tumor growth and non-tumor growth when the cells were treated with an LNP. And this happens relatively quickly. So within 72 hours, the tumor seems to start receding. In this particular case, and I'm not exactly sure how this is going to play out, it looked as if the disablement of NRF2 at R34G, this is a unique site, actually stopped the tumor from growing as well. But we do feel that this is just a little bit of optimism there. We think that we'll have to come back and treat with chemotherapy or radiation going forward. A couple of other characterizations. I was trained as a molecular biologist and a nucleic acid biochemist. And so not only do we want to see that the tumor size was changed, we extracted the tumor at endpoint, and we looked at the level of NRF2, which we can see on the far right-hand panel is actually down significantly. And that's also been confirmed by Western blot. But also the Ki-67, which is a measure of new replication fork initiation. So that's really an important one, is replication fork initiation. And that has basically been installed in cells that are treated with NRF2 after a period of 72. These are the kind of details that are very important in oncology to measure. It's not simply size of the tumor, that's not good enough. It's good, you have to take the tumor out and make sure that the cells in the conjunction of the tumor, the complexity of the tumor, have actually stopped growing. Took this to the FDA, and they seem to be very excited, very encouraged. The FDA in the United States doesn't ever really get excited or encouraged. They can't give you that kind of advice. You have to prompt questions from them, and they were really, really supportive. But they also decided to push us a little bit forward to expanding this to a basket approach. So not just lung cancer, and the squamous carcinoma of the lung exists at the top of the trachea, or at the bottom of the trachea. They also felt that this would be effective against head and neck cancer and esophageal cancer. So just to review a little bit about head and neck cancer. One of the things I mentioned at the beginning is we wanted to make sure that our technologies, which we hope are more simple-minded to some of the other effective ones so far, would also have a chance to reach underserved populations. And head and neck, believe it or not, is one of the most ravaging cancers in lower economic social groups. Why? Chewing tobacco, alcohol, and smoking exacerbate this problem a lot. It is endemic across the United States and the South. So sort of from Georgia all the way across to Wyoming and Oklahoma. And Oklahoma has actually come forward as a place that we hope to be able to treat patients initially. So it's the 7th most diagnosed cancer in the United States, probably surprised at that. And 90% of squamous cell carcinoma were our original animal models were done in. We think about 30% will increase dramatically. And that's a little bit of a surprise, but again, smoking and alcohol consumption, chewing tobacco tend to be endemic in the whole country, but it is pocketed in the United States. And it's these people that rarely receive breakthrough therapies, very rarely receive them. So we want to be true to the mission, understand about the raising of money and things like that. But I listened very carefully to what Jimmy was saying yesterday. Don't just put it on your website, implement it, disseminate it. And so that, fortunately, is what our approach has been. So we did a standard series of experiments in tissue culture and in animals, and not surprisingly, we were able to show increased sensitivity as a function of addition of cisplatin, which is one of the chemotherapies used for the treatment of head and neck cancer. Monotherapies include Keytruda, plus followed up with radiation or chemotherapy. So head and neck cancer is traditionally treated with a combinatorial approach. So that's been really, really exciting. The animal data was actually really interesting, but I did want to show you a paper from a Japanese group that had a very interesting outcome. So they did the same experiment we did, except in head and neck cancer. They knocked NRF2 out, and they showed an extreme reduction in tumor growth by itself again, not with adding chemotherapy. Again, we're hoping this is true. Our data suggests you kind of need the secondary treatment, but at least conceptually now we have a second group that is shown, and that slide there of the tumor should look familiar to the ones that I showed you previously, roughly the same experiment. And then a good colleague and a good friend from Israel, Dan Peer, just published a paper where he showed that 15% knockout of a gene known as SOX2, we work on SOX2 as well as NRF2, essentially obliterated tumor growth by itself. This is an astonishing result. Dan is a great guy. He's an expert in lipid and LNP delivery. So his data mirrored what we were seeing. How is this possible? How is it possible that 15% of the knockout of these transcription factors can give you such a dramatic approach? We haven't gone out 84 days. We've only gone out about 60, but we anticipate our tumors staying roughly the same. Why is that the case? It comes back to how the gene is chosen. So here's an interesting experiment. So here's where we looked at the amount of editing versus chemosensitivity at 3 days, 7 days, and 17 days. The gray bar, the darker gray bar, shows you the percent of editing we get in cells. Amazingly, right, about 85%. As time goes on, that decreases because the wild-type cells start to grow back, which is the common problem in cancer, except the NRF2 is barely expressed, which allows it to, those cells, even in the wild-type zone, allow them to be susceptible to chemotherapy. Remember, those cells are aggressive because there's high levels of NRF2 in them. So as they start to repopulate the tumor, the chemotherapy can be effective because the threshold levels of NRF2, at least in this 2 to 3 week period, have not been reached. In Pierre's group, they saw it out to about 2.5 months. So I think we're both on the same path. I'd like to believe his data more than me, but at the moment, we'll stay a little bit more conservative. So here's what we think happens, okay? NRF2 is a really critical gene because it's not just your normal knockout. I see people knock out a particular gene, they get 80% knockout, and they think this is great. Well, knocking out a transcription factor that controls downstream activities is actually a little bit more effective as long as you choose the right outcome. So with drug resistance, if you knock out 15 to 20% of the NRF2, either by our method or by others, NRF2 or SOX2, you do see complete cessation of the tumor growth. To us, this is really promising, very encouraging. Obviously, a lot more work to do, but we're encouraged. Here's something about head and neck cancer, esophageal cancer, and some forms of lung cancer. These tumor cells develop what's known as the NRF2 addiction. So in addition to the types of responses I showed did NRF2 controls, tumor cells in head and neck convert themselves into energy dependent via NRF2. So if you disable NRF2, what happens is the level of energy production starts to diminish. The tumor cell then, at least in our hands, is much more susceptible to the secondary treatments of chemo, radiation, and now we believe immunotherapy. So this gene is special, and it's special in about 3 or 4 solid tumors. If it's not upregulated, this is going to be a little more challenging, but it still could be effective. It doesn't matter if the NRF2 is protecting a tumor cell anyway. But for the NRF2 addiction, and the following thing, which is now emerging very quickly, is that NRF2 deactivation increases the immune response inside the tumor. So we have a little bit of data on this. I do know some other data that's coming out. So in the long run, if we're able to re-emerge, restructure the NRF2 levels in a tumor cell, then hopefully we'll be able to activate these innate TIL cells, which we heard such a great talk of this morning. And it's all about following the data. This is one of the challenges in gene therapy and in gene editing because money follows either big names or the right zip code. But in terms of following the data, this is where we seem to be headed. Now, how do we get it there, right? So we're using a non-viral LNP, and using surgical oncologists, we will directly inject the CRISPR into the tumor. Now, direct injection is really specialized for tumors you can see. And in head and neck cancer, esophageal cancer, the metastatic tumors are also accessible by injection. So our clinical team, Barbara Burtness from Yale and Glenn Hanna from Harvard, have all been assembling a clinical protocol that should allow us to do that kind of direct delivery. I hope at some time, and I'm hoping to hear later in our delivery sections, that we have developed LNPs that can specifically target certain kinds of tumor. That's the solution. But the amount of stuff that has to go into the blood, even with the Intellia work and liver, is pretty seriously high, and people do get sick from that. By direct injection, we kind of avoid sort of those side effects. And then finally, just want to talk a little bit about a small company that emerged from the Cancer Center. So we actually started to believe this after about 5 or 6 years of really, really hard work. And eventually, combining with the NCI Center of Excellence at Christiana Care Cancer Center, we formed a spin-out company called CRISPR. And that was really only not so much to commercialize a product, but actually to advance it through the FDA and onto potential clinical trials, where we see the glass as cautiously optimistic, cautiously half full. And here's what's interesting about this approach. It's pretty agnostic to the downstream treatment. So we could help radiotherapy, we could help chemotherapy, and we also believe now we can help immunotherapy approaches, because it's essentially giving another tool to the surgical oncologist or to the medical oncologist who can just use to inject it directly in the tumor. Most importantly, surgical oncologists want to cut the tumor out. So initially their response is going to be to inject, wait 72 hours, reduce the tumor. And the end point of this, believe it or not, is likely a lowering of the amount of normal tissue that has to be removed from the body. That's the real key. So we're on the clinical verge of that. These endpoints are amazing. They're very limited, and they almost seem like low-hanging fruit. And then finally, I just want to give a shout out to all the partners who have joined us. CCure just signed last week. They'll be doing the off-target stuff. We've actually done off-target analysis, not only in nomination, but in validation phase. And if you want to kill yourself for a year, try doing that alone. And I want to shout out to IDT that gave us a lot of support on that. They sort of saved us. The manufacturing group is in place. Dan and her team is going to manufacture the drug. We're underway. And importantly, Precision for Medicine, which is a great group of people that work primarily in developing clinical trial structure and have been really key in getting our key opinion leaders on board. And then finally, just a shout out to the patients. More than likely, this trial will initiate at a place that people wouldn't think of, and that's at the Oklahoma Health Center, Science Center. A large number of people, including Native Americans, actually suffer from head and neck cancer because of the smoking and the alcohol abuse. So this may seem like a really off shot from what we normally hear in the great places that do this, but I'm hoping we can stay true to our mission and continue to move forward. I had a chance of talking to you today. These are the people who've done all the work. Thank you very much.
Thank you very much, Eric. Great talk. Yes, please. Yeah?
Well, thanks for the great talk. 2 questions, short one. The NRF2, is it expressed more highly on the mRNA level, or on is stability of the protein?
That's a fabulous question. It's mRNA.
It's mRNA.
Yeah.
Okay. And second thing is, if you would target NRF for cell depletion, would that help in the same way like cisplatin?
For what, sorry?
If you just target NRF and you would have a tool to deplete the cells by the expression of NRF2, would that help as well?
Yes. So multiple targets have been developed. So NRF2 has been a very appealing target for companies like Bayer for years. And where we are is they have nutraceuticals that use NRF2. The reason for that is the structure of the protein is floppy. And so there's a whole unsegregated region that tends to turn. So small molecules have not been effective there. The original observation for using NRF2 with chemosensitivity came in 2008 from a woman named Donna Zhang at Arizona State University. She did siRNA, knocked it down, and then was able to see the chemosensitivity. So the problem is that the turnaround is really quick, because in a hyperactive one, you have a lot of mRNA in there, so it's hard to knock it down sufficiently. But the original idea came from Donna Zhang.
Would appreciate a discussion afterwards.
Mm-hmm.
Hi, great talk. Sorry, a question from my side. I would be very curious whether you envision that this treatment may actually then lower the all immune therapy and chemotherapy that patients have to go through, that you explained, that actually may also be a reason for why they either drop off or they suffer from side effects that eventually drop off from a treatment.
Yeah, that's it. And again, I realize it seems like a very low-hanging fruit goal, but because our mission and our baked in is to reduce the suffering of patients, this is what patients tell us they're going to use. And from a commercial standpoint, and I think it was Jimmy or someone else said this yesterday, patients don't want to wait for huge regulatory approvals. They want access to treatment. And that's what we find too, is that they just want to feel better. And as chemotherapy goes on, and despite our great effects, and we hate chemotherapy, we all know that, still about 70% of lung cancer patients and head and neck cancer patients default to chemotherapy. They'll start with immunotherapy, and some of it's very successful, and it's great news and great advances. But ultimately, if you can just keep the patients healthier for a period of time, and even better drugs can come forward. There's some argument we actually have enough cancer drugs. They just don't work effectively enough, or they really hurt the patient. So in our world, thinking about how we can use a very simple-minded approach in solid tumors that we can access. So this is not going to work in pancreatic cancer. It probably won't work in colon cancer because it's, the amount of drug that you have to get there effectively will be high. So by direct injection, we can. So I think the first target will probably be oral cavity cancer of the tongue. That's a major problem with smokers. So, yeah.
So direct injection into the tumor is a nice way to avoid some side effects. But how many cells do you actually reach? And how evenly do they get?
Great question. So, it's hard to model head and neck cancer in the tongue of a mouse, but it actually can be done. And so we get about 70% penetration. So the distribution inside the tumor is not so much the problem, but the question I think you're leading to is, and that is, what size tumor do you go after? Okay. The tongue cancer starts with a white dysplasia. So I know everyone's going to go in the bathroom and see if they have a white patch on their tongue. And that can be either malignant or benign. Most of the time they go away, especially if you smoke. You'll see these come and go. So what the KOLs are currently doing is to decide what the patient population is in that. I'll tell you the bigger problem, to be honest with you, with surgical oncologists. They want this one time. They see it as a neoadjuvant, inject it, wait 72 hours or 2 weeks, and then remove the tumor. Because what happens is that the chemotherapy and radiation alone in the mouth and the head area, even if you start with Keytruda or another immunotherapy, is so severe that people start to burn holes in their cheeks and their pharynx and in their tongue. So the size of the tumor is currently up for debate, but in the surgical oncologist world, it doesn't matter. 30 microliter injection into the tumor, no matter what the size is, will hopefully reduce the tumor with very little side effects, and then the chemotherapy can start after that. So.
Okay, there's, yep.
Thank you. I want to ask, NRF2 is known to regulate quite a number of enzymes to balance redox. So I don't know, disrupting NRF2 because of cancer, are you sure you're not going to create another problem elsewhere in balancing redox, restoring redox balancing?
Excellent question. And the answer is that if the effect of knocking NRF2 out is much more impactful in NRF2 cells, in cells that have elevated levels of NRF2, okay, and that's a tumor cell. But that is exactly why we believe in localized delivery, direct delivery, not in systemic delivery. We love to inject it systemically. Now, you could argue that our tumor-specific target, and we found this in melanoma, there are plenty of, this is pretty well known through the field, KRAS, NRAS, NRF2, I think EGFR, we found unique sites that are tumor-specific targeting. But how do you deliver that specific molecule to target that tumor? And injection systemically, to me, seems still a bit out of the world. So you are going to lose some normal tissues, normal cells that are baked into the tumor, no doubt. They will also be knocked down, but probably not as effectively as those that have NRF2 levels that are upregulated. So it's really all part of the delivery process. And, you know, intratumoral injection for head and neck cancer is fairly common. It's not loved. People want to be injected. But if you ask patients who would much rather go through that once every 6 months, perhaps, or once every quarter, as opposed to weekly infusion. I think Oskin said this morning quite nicely, that patients have to get this too. So his work is essentially like ours. We're trying to reduce the amount of times and pressure they have to go through. We also hope long-term this is a little more cost-effective because it's a synthetic drug that gets directly injected there and won't have to require all the pre-treatment sort of stuff. So.
Great. I have a quick question over you, Eric. Great talk. So I was wondering whether in the preclinical studies and in the clinical studies you follow the clonality, how many of the cells had the biallelic frameshift mutations, and what was the selection advantage following time?
Let's see. So in the, yeah, I don't know the answer to that. Let me think about that directly. What we've done is when we first did the knockouts, we isolated probably about 400 clones and grew them all up and then tested them. And the sensitivity seems to be about the same, but it did depend on the type of the indel that's created there. So we are dependent not only on knockout efficiency, but we're dependent on the type of indel that's created, particularly a frameshift. And I think that's why Dan had such extraordinary results with SOX2, is he created a very proprietary LNP, which not only delivered it very effectively, he did intratumoral injection. It's the same experiment we've done, exactly the same. He beat us to publication, but he's a great guy and a really reputable lab. I have a lot of respect for him. And I think what his system did is it created this indel code, this higher number of frameshift mutations. Now, in clonality escaping, I don't know, the tumors in the mouth are vascularized. It's hard to recapitulate that in a mouse. So if some clonal expansion occurs unpredictably, we're not going to know. By direct injection, the other advantage is the biodistribution is amazing. It's about 99% in the tumor. We don't really see much else going other places. But time will tell, when we get to that.
So thank you very much, Eric, for a great talk. And now Eric is going to take it away from here and continue as the chair of the session. Thank you.
My pleasure. Okay, so our